AP Bio Spring Final Project. In this presentation, I will be talking about Topic 6.5 and 6.6. Topic 6.5, Regulation of Gene Expression. Contents of this topic: the types of interactions that regulate gene expression, including regulatory sequences, epigenetic changes, and the determination of the phenotype of a cell. Location of regulatory sequences in relation to their function, including prokaryotic and eukaryotic groups that are coordinately regulated. Gene expression is the process by which instructions in the DNA are transcribed and translated into a functional protein. Regulatory proteins bind to specific sequences in DNA in order to control which genes turn on and when they turn on. Regulatory sequences are segments of DNA that work with regulatory proteins to increase or decrease the expression of specific genes. Epigenetic changes are changes that are reversible and do not change the DNA sequence, only how the body reads the DNA sequence. Histones are proteins that associate with DNA in the nucleus of a cell to help condense the DNA into chromatin. Phenotypes are the observable characteristics of an individual. Transcription factors are proteins that control the rate of transcription of DNA into an mRNA and overall regulate gene transcription. Cell differentiation is the process in which a cell changes from one type of cell to another. The steps of gene expression. Step 1. Transcription is the first step of gene expression, in which a segment of a DNA strand is copied onto a new strand called an mRNA. Analysis. During transcription, it is up to certain molecules to decide what to do specifically in order to result in the desired product, which regulates the gene expression. How is gene expression regulated? Regulatory sequences. These sequences, such as the TATA box, are located on every DNA strand. When regulatory proteins, such as repressors or activators, bind to the regulatory sequence, gene expression is either prevented or promoted. Repressors either prevent the RNA polymerase from binding to the DNA or just slow it down in the transcription process. Activators enhance and help the transcription process. Epigenetic changes. There are epigenetic changes such as DNA methylation, which changes the activity of DNA but not the sequences, and histone modification, which can cause the histones to condense the DNA in different ways. The way the DNA is packed by the histones, either tight or loose, contributes to the expression of the gene. Therefore, these epigenetic changes can change the way DNA is read and alter the accessibility of the DNA, overall controlling gene expression.
What is genetics? Wait, I've already done that one. What is epigenetics? Genetics is your genes. A gene has a unique sequence to make a protein that does something in your body. A genetic mutation is a change in one or more parts of this sequence. This could just make you, or it could contribute to a genetic disease. An epigenetic change also changes a gene's DNA, but not at the sequence level. Instead, special marks are added or removed that might change how a protein works in the body. And this could also contribute to disease. How these marks appear is a hot research topic, but some causes could be diet, stressors, or environmental pollutants. A genetic mutation is difficult to fix. With epigenetics, we could take a different approach to treating disease by finding ways to remove bad epigenetic marks or add good ones.
How epigenetic changes regulate gene expression. Methylation of DNA histones causes nucleosomes to pack tightly together. Transcription factors cannot bind to the DNA and genes are not expressed. Histone acetylation results in loose packing of nucleosomes. Transcription factors can bind to the DNA and genes are expressed. What does gene expression affect? Phenotypic determination. Phenotypes are the expressions of a gene just in a way that humans can see them from the outside, such as hair color and eye color. Phenotypes are the physical representation of genotypes, which are determined by the combination of genes, dominant and dominant, dominant and recessive, etc. The level at which genes are expressed also contributes to what the phenotype will be, because some genes are not fully expressed, resulting in different phenotypes. Cell differentiation. Sometimes, genes that code for tissue-specific proteins are translated and transcribed, resulting in those tissue-specific proteins. When these specific proteins are synthesized in a cell, the cell turns into a specific type of cell correlated to the type of tissue-specific proteins it produces. For example, a normal cell experiences cell differentiation when it produces muscle proteins, such as actin and myosin, and turns into a muscle cell. This observable cell differentiation is a direct result of what types of genes are expressed. What affects gene expression? Transcription factors. In order to make sure gene expression goes smoothly, transcription factors are inducted. Transcription factors bind to DNA to turn genes on or off, and this is very necessary because it ensures that the correct genes are expressed at the right times in the right parts of the body. In this example, there are some activators and some repressors. The gene is only expressed if both activators are present and the repressor is absent. So if both activators are present, transcription will occur, and if one activator is present, there will be little to no transcription. However, if the repressor is there at all, there will be absolutely no transcription. Regulatory sequences. Location and function. Regulatory sequences, such as enhancers and silencers, are located in specific regions based on what they are meant to do. Some regulatory sequences are located close to the promoters that they regulate, while some are further away. The location of the regulatory sequence relates directly to its function. For example, promoters, which are a part of regulatory sequences, are usually located at the 5' end where transcription will begin, because the function of a promoter is to turn the gene on or off. Promoters have a binding site where RNA polymerase will start transcription, which is why the promoter sequences are located at the beginning. Coordinately regulated genes in prokaryotes. Operons, sequences of DNA containing a cluster of genes under the control of one promoter, allow protein synthesis to be regulated coordinately based on what the cell needs at that time. Operons are produced from multiple genes that are transcribed together, resulting in one mRNA that is then translated into an operon. Operons contain regulatory sequences that are binding sites for regulatory proteins that promote or inhibit transcription. An example of an operon is the lac operon, which encodes proteins that allow prokaryotes to use lactose for energy. When energy is low and lactose is present, the operon will be turned on and start using the lactose. Due to the fact that the operon is off unless lactose is present, it is an inducible system, because the system is off unless an inducer is present that allows for gene expression. Here is the lac operon. The first example is when there is no lactose. When lactose is absent, the lac repressor binds tightly to the operator. This gets in the way of the RNA polymerase, therefore transcription cannot happen. The second example is with lactose. Allolactose, which is rearranged lactose, binds to the lac repressor, which makes it let go of the operator. RNA polymerase can then transcribe on the operon because there is no repressor in the way. Coordinately regulated genes in eukaryotes. When genes in eukaryotes are coordinately regulated, they can be done so by the same transcription factors. Coordinately regulated genes are located on the same chromosome and can be activated by the same factors. There are no operons in eukaryotes, and each gene is made of its own mRNA, but they can still be affected by the same factors. Topic 6.6: Gene expression and cell specialization. The contents of this topic are how the binding of transcription factors to the promoter regions affects gene expression and/or the phenotype of the organism, which includes that promoters are DNA sequences where transcription is initiated, and negative regulatory molecules inhibit gene expression. And the connection between the regulation of gene expression and the phenotypic differences in cells and organisms, including that gene regulation results in differential gene expression and influences cell products and functions, and that certain small RNA molecules have roles in regulating gene expression. Transcription factors binding. What happens? A promoter is a sequence of DNA where transcription is initiated. The promoter contains a binding site where RNA polymerase can bind and begin transcription. Transcription factors identify the specific DNA sequence in the promoter and bind to it. Then the RNA polymerase binds and transcription occurs. What does this affect? Transcription factors are always working to regulate gene expression by boosting transcription or turning genes on or off in certain parts of the body. For example, activators boost gene expression by signaling the RNA polymerase to continue past the promoter and move along the gene. Since phenotypes are the visible expression of genes, if transcription factors affect gene expression, they also affect the phenotype of an organism. Negative regulatory molecules. What do these affect? Negative regulators are elements that inhibit gene expression by blocking the enzyme that transcribes the gene. Repressors, for example, bind to the gene and either prevent the RNA polymerase from binding to the promoter, or they just slow it down. This negatively affects gene expression because the genes cannot be expressed if they are not transcribed. Results of gene regulation and what it influences. Differential gene expression, or DGE, and cell products / their function. DGE is the idea that every cell in an organism has the same DNA but expresses different parts of the genome depending on what type of cell it is. In cells, not every gene in the DNA is made into a protein because not all cells need that specific protein. When genes are regulated, what genes are expressed and to what extent is controlled results in DGE. The specific type of cell that a cell is influenced by gene regulation because it controls what genes are expressed. The combination of expressed genes is what determines the function of a cell. Small RNA molecules, what do they do? Certain small RNA molecules that are 18 to 30 nucleotides long and non-coding, such as RNA fragments, can have a role in gene regulation. For example, these small molecules can block transcription by binding to the mRNA, cause degradation of mRNAs by base pairing with complementary sequences and facilitating cleavage, increase the expression of target genes through RNA activation, and more. Gene regulation and phenotypic differences. How do they connect? Phenotypes are how we see gene expression. The combination of genes that are expressed is what gives us our phenotypic differences. Without gene regulation, the body would have no way of controlling what genes were expressed, where they were expressed, and when. This would likely result in organisms that most likely could not function at all, organisms that had only one type of cell. Gene regulation is necessary for life, and it is what makes us so different and unique.